Hello everyone. Today we're going to be talking about microbial metabolism once again. We'll be focusing on cellular respiration, oxidative phosphorylation, and fermentation. Remember that organisms that are aerobic are going to use oxygen to make their ATP during the processes of cellular respiration and oxidative phosphorylation. So far, we've only discussed the fact that oxygen's role in these processes involves accepting electrons at the end of a long series of reactions. Anaerobic organisms are going to use a different molecule while they're making ATP during cellular respiration and oxidative phosphorylation. They're going to substitute in a molecule such as nitrate, or maybe sulfate, or fumarate, or carbonate, some other molecule other than oxygen that can serve as a final electron acceptor. Remember, most bacteria are facultative anaerobes, and what that means is they are capable of both aerobic and anaerobic cellular respiration and oxidative phosphorylation. So let's think about these terms that we're starting to discuss in detail today. Let's think about exactly what processes we're talking about. Cellular respiration is a series of reactions that breaks down or oxidizes a simple sugar, such as glucose. Remember, glucose is a 6-carbon molecule, and by the end of cellular respiration, all of the bonds will have been broken down, so that the only thing that's left is a 1-carbon molecule called carbon dioxide. As we've said, cellular respiration can either be an aerobic process involving oxygen or an anaerobic process involving some other molecule besides oxygen. Oxidative phosphorylation follows cellular respiration in both aerobic and anaerobic organisms. Oxidative phosphorylation uses the energy that is harvested during cellular respiration, as well as a structure called an electron transport chain, or an ETC, as well as a hydrogen ion gradient, and a critically important molecule called ATP synthase to build this molecule we call ATP. Oxidative phosphorylation follows cellular respiration, so it may follow the aerobic form of cellular respiration or the anaerobic form of cellular respiration. Oxidative phosphorylation has nothing to do with oxygen. That word oxidative is not related to the word oxygen. Instead, it's related to the word oxidation, as in oxidation reduction reactions. Finally, there's this process called fermentation. During fermentation, we're going to see anaerobic metabolism of glucose intermediates, or the intermediates from other simple sugars. And those intermediate molecules will be metabolized to produce certain product molecules while regenerating what are called electron carrier molecules. Along the way, a small amount of ATP will also be produced. Now, fermentation is the primary metabolic pathway for some microbes. In other words, that is their primary preferred form of ATP production. But in many other microbes, fermentation is a secondary metabolic pathway. It is a process that's going to occur only when the microbe is unable to perform cellular respiration and oxidative phosphorylation. In other words, fermentation only happens when those organisms have no access to their final electron acceptor molecule. Like human beings, many microbes are going to make their ATP using cellular respiration and oxidative phosphorylation. Let's think about all of the different reactions that are happening during those two processes. In summary, during cellular respiration, microbial cells and other types of cells are going to take a glucose molecule and they're going to break it apart, bond by bond by bond, until that 6-carbon sugar is broken down into a 1-carbon molecule called carbon dioxide. Now, all living cells can break apart glucose. In other words, all living cells can use glucose in the process of making ATP. Some microbes are able to use other simple sugars as well. For example, we talked about the medium called mannitol salt agar, and we said that some organisms, specifically Staphylococcus aureus, is capable of fermenting a sugar called mannitol. Staph aureus can use glucose and does use glucose in its metabolic processes. It's just also able to ferment mannitol under different circumstances. We also talked about a medium called EMB, eosin methylene blue agar, and we said that that agar contains lactose and sucrose. And by looking at the color of the colonies that grow on EMB, we can tell if those organisms are capable of fermenting lactose and or sucrose, which are two other simple sugars. So yes, all organisms use glucose for energy, but some organisms are also able to use additional simple sugars. So, during cellular respiration, cells are going to break apart glucose or another simple sugar until they've created a 1-carbon waste molecule, carbon dioxide. Along the way, as all of those covalent bonds are being broken, the electron pair that's in that bond will be released. Remember, electrons are the conduits of energy. So when a bond is broken, a covalent bond, and the pair of electrons that sits in that bond is released, energy is also released. The cell is going to harvest the energy from those electrons in a structure called the electron transport chain, or again, the ETC. And after the electrons pass down the ETC, they will be passed on to the final electron acceptor molecule. So that is a basic summary of cellular respiration. Breaking apart this sugar and harvesting the energy that is released through the breakage of those covalent bonds. Now, during oxidative phosphorylation, the energy that was harvested from the electrons in the electron transport chain is going to be put to use. The energy is going to be used to build a gradient, specifically a hydrogen ion gradient. And remember, in microbial cells, ATP will be produced at the cell membrane. So this hydrogen ion gradient is going to be created across the cell membrane. Now, what do I mean by that? Remember, a concentration gradient is simply a difference in concentration across a membrane. So that means that on one side of the membrane, there'll be a relatively low concentration of hydrogen ions, and on the other side of the membrane, there'll be a relatively high concentration of hydrogen ions. So there'll be a gradient of hydrogen ions. Now that ion gradient is going to be allowed to resolve or go away by passing the hydrogen ions across the membrane from the side with the high concentration toward the side with the low concentration through the center of a molecule called ATP synthase. The hydrogen ions will literally flow across the membrane by flowing through a tunnel in the center of this molecule called ATP synthase. They will continue to flow down the concentration gradient until equilibrium is reached. And along the way, ATP synthase will manufacture ATP. Cellular respiration and oxidative phosphorylation are then a long series of reactions. Cellular respiration and oxidative phosphorylation are actually a special kind of series of chemical reactions known as oxidation reduction reactions. We often refer to these types of reactions as redox reactions for short. Now, oxidation reduction types of reactions are always paired with each other. So if during a chemical reaction some molecule gets oxidized, that means some other molecule in the reaction must be reduced. If during the reaction some molecule gets reduced, that means somewhere else another molecule is being oxidized. The two reactions are paired. They always occur together. Oxygen and the other molecules we've talked about that can be involved in cellular respiration are the final electron acceptors at the end of this structure called the electron transport chain. We're going to be removing electrons from covalent bonds. They will be released as the covalent bonds are broken in glucose or some other simple sugar. So because the sugar molecule is being degraded, because it's being catabolized, remember, the breaking of bonds releases energy. We said that was exergonic. That also can be thought of as the glucose molecule being oxidized. It is losing electrons as its bonds are being broken. Now, from the start of the glucose being broken down to the end, where there's carbon dioxide left, that's a long series of reactions, and they are oxidation reduction, oxidation reduction, oxidation reduction in a long series. We start by breaking that first covalent bond in the glucose. That's an oxidation. And we're going to finish way at the end when oxygen, as the final electron acceptor, receives electrons and becomes reduced.